Welcome everybody. Here we'll discuss about new double mutant or triple mutant coronavirus, which is called Indian variant, and which is over 70% in Indian coronavirus patient. Here this is called B.1.617. Let's know that what is actually the double mutant and the triple mutant, whatever it is, the meaning of it and what it's different from the previous coronavirus. Okay, we can start. Here, this is the this is the double mutant or triple mutant. Triple mutant that B.1.617, the variant. It is also written as the variant under investigation, BUI. It is just a variant of in COVID virus. It is not the new virus. Variant, it means that some mutation takes place. Mutation, suppose here some changes will occur, which is here it is shown in the black, and here I have shown the red. It means that something different, and show here it's some changes in that in spike protein. It may occur. Okay, so this is called antigenic drift. And this is new variant, I mean in gross that I have shown. Now we are talking about the double mutant or triple mutant coronavirus, but actually this virus which we are talking about, that B.1.617, is 13 mutation is recorded, not 1, 2 or 3. This 13 mutant, but we are calling as double or triple. The why so? We can understand that. Okay, any virus having its other different part, with that the spike protein outer covering the nucleocapsid, and with that their genetic material. But our interest is clinically, this part is important, that is the spike part, spike protein. Spike, you can understand, this is the virus and here is the spike, and this is the attachment zone, which is called RBD, receptor binding domain. So in this is the attaching point to the human body, the lungs. So this area is our interest, and inside other part it is not. Look at here you can see that the area where from here to here, where is the viral genome, and where the changes, I mean mutation will occur, that is it is here. Open reading frame, this is called open reading. Open reading frame means that there is no stop codon is there, and the viral genome, exon part is there, so gene is there. So there is some changes here, in that way the open reading, another one open reading, which is viral enzyme that can makes. Now come here the spike part. There is 4 mutation. See that here is 1, 2, 3 and 4. This last 2, it is written in big letter, because these 2 changes, it makes severe changes in that variant. Other changes is not so. So that is why these 2 are very important, and what changes are there, that I will tell you. And other part you can see that another ORF, open reading frame, and this is the nucleocapsid, some changes are there. Total if you count, that will be 13, but our importance is 2 here, so that is why it is double variant. If you include this one, then it will be 3. So what are these two? That E484Q and L452R. If you include the third one, D614G. Now, what is the meaning of that? Let's learn a little bit. This meaning is AL here, it indicates that which is found in the previous COVID. Yeah, this amino acid is found in the previous COVID virus, but it changes into some other form. Here R, what is AL, what is R, I'll tell you afterward. And in between one certain number, in every cases you can see that every cases this number in between these two English letter, that is suppose here 452 is that nth of amino acid. In that particular amino acid, this changes is there, that it indicates. Okay, so this name is quite scientific, that you have to understand. Later we can discuss this three mutation. The first one here, that E stand for here glutamic acid, which was found in the normal wild type of COVID. It changes into glutamine. So amide, so acid to amide, it changes, and 484th amino acid, this changes is there, and this changes the virus made for the escape from the vaccine, and for that this is called the escape mutation. So in that way the virus efficiency is decreased 50%. So this one is clinically very important. I think everybody got that. And the glutamic acid which was there and now not there, instead of glutamic acid, glutamine is there. In another changes in spike protein, that L, which is a leucine hydrophobic amino acid, which changes into arginine, positively charged amino acid, and this changes makes that 20% more contagious. So now the new viruses, but it may be less virulent, but still research is going on, so we cannot say all this confirmedly, but it is contagious, that is confirmed. Elution, it converts into arginine, in which amino acid? 452nd amino acid. The third one that we don't know more about that, this the here is aspartic acid changes into the glycine in 614th amino acid. Here the problem arises in that antibody which is prepared by the by our body, which can be through vaccine, can be natural way. In both cases the problem occurs. Problem occurs because in normal variety it was in RBD, the receptor that binding domain, which will binds with in human body the lungs, ACE2 in lungs. So it contains, previously it contains leucine, glutamic acid and aspartic acid, but now it is glutamine, arginine and glycine. So antibody will not detect all this. It is almost like that to catch a criminal, you have given the photograph to the police, to find out that person, but in the meantime that criminal changes his face. So how the police can detect this? Almost like that kind, and the affinity of that, it is increased in that way. So all these are that I have discussed, that I think you'll get some information from that. Thank you.